Hello students, welcome to Biodimension online classes. From today onwards, we are going to start a lecture series based on enzyme. And today, we are going to start with the basic concepts of enzyme. We all know that enzymes are catalysts of biological systems. That means enzyme we actually use or our body, our system use enzyme as a catalyst and it's a biological catalyst. Enzymes are remarkable molecular devices that determines the pattern of chemical transformations. Yes, whatever the biochemical transformations are continuously occurring within our body, that biochemical transformation are actually based on the activity of these enzymes. They also mediate transformation from one form of energy into another. We all aware of our energy generating systems within our body, not within our body, from starting from bacteria to human. In each and every organism, whatever the energy generation process are, the biochemical pathways are, like glycolysis, TCA, oxidative phosphorylation, electron transport chain, all these biochemical pathways are actually based on the activities of enzymes. So, in this biochemical pathway, what the enzymes are actually doing, they are actually converting chemical energy into the ATP or the heat energy, which is required for our body. About a quarter of genes in the human genome encode enzymes and evident to their importance of life. So that is very important part of a genome. So if we consider the genome as a director of our body, the main worker line molecules, main worker line biomolecules, which molecules are actually performing the basic work within our body is the enzymes. So that is why nearly a quarter of human genomes, a quarter of human genome is encoding the enzymes. The most striking characters of enzymes are their catalytic power and specificity. Enzymes are very specific in their nature, specific towards substrate. We will come into that thing later. So the catalysis, the reaction, the catalytic reactions takes place at a specific site on the enzyme. And that specific site is called active site. Now, if we look at the functionality of protein, enzymes mainly composed of proteins. Some of the enzymes are also available. We'll come that for later. But the proteins are the main building blocks of our body, main building blocks and main performing molecules. We have other biomolecules also like lipid, like nucleic acid. Each and every molecule have their own role, but protein have multiple roles. And it's starting from the structure and mechanics, whatever the muscle we have, that is all composed of proteins, hormones, signaling molecules, whatever the signaling molecules, that means the signaling molecules are also composed of proteins, antibodies, which is a very important part of our immunity, fluid balance within the body, and that actually uh, 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 caused by the different proteins which are available like albumin, globulin, and all that. Acid-base balance within our system, channel and pumps and transport. All of these actions are actually conducted by proteins. Also, protein have a very important function they are actually acting as or working as enzymes also. So mainly, nearly all known enzymes are protein. Nearly all known enzymes, whatever the enzymes we know, like starting from the very beginning, like amylase, like other enzymes like DNA polymerase, RNA polymerase, like cellulase. These all enzymes are protein. However, protein do not have an absolute monopoly on catalysis. So, that doesn't mean that proteins are the only building blocks of enzymes. Some of the enzymes are also available, like the catalytically active RNA molecules, RNAs, what we call. We will do a separate lecture on the RNAs also. But the RNAs or catalytically active RNA are also a very good biocatalyst, which was actually the biocatalyst early in evolution. Now, why protein? Why not any other molecule? Protein as a class of macromolecule are highly effective. Highly effective like what? Highly effective as catalyst for an enormous diversity of chemical reactions. So proteins have the ability to catalyze the enormous diversity of chemical reaction. They have a significantly very important structural specificity also. So they are able to bind a wide range of molecules. That is basically because of their structure. By utilizing the full repertoire of intramolecular forces, by utilizing enzymes being substrate together in an optimal orientation. So enzymes need to bring its substrate much more closer and use the catalytic part of the protein to break chemical bonds and to form new chemical bonds. These reactions need to get stabilized and need to cross their energy transition states. That is why protein are able to bind with their substrate in a very close manner and able to perform all these kinds of reactions. So by selectively stabilizing a transition state, an enzyme determines which one of several potential chemical reactions takes place. Several potential chemical reactions, just like breaking a covalent bond, just like oxidation reaction or a hydration reaction. We know several kinds of reactions are there, and based on each and every kind of chemical reactions, we can divide enzymes into different categories. So that is why proteins are very much important, because they are able to select, they are able to determine that which of the chemical reactions need to take place. They have the capacity to control, they have the capacity for control. Control of what? The catalytic activities of many enzymes vary in response to the concentration of substances other than their substrate and product. The mechanism of this control process include allosteric control, covalent modification of enzyme, variation of the amount of enzyme which is synthesized. So in our body, we a particular reaction need to get stopped at some point of time, and the enzyme being a protein, we can the body or the system can able to regulate that protein by interfering with their structures. And that is why allosteric controls come into the picture with the other control like the covalent modifications and like that. So that is why the system, the biological system, is able to control the enzymes in such a way which is not possible with the other biomolecules. That is why enzymes have proteins. So what are the difference with the chemical catalysis? So differences with chemical catalysis. In which way the enzymatic catalysis differs with the chemical catalysis. So here it is not actually 10 to 106, it is 10 to the power 6 and 10 to the power 12. The higher reaction rate, the reaction rate is higher. The enzymatically controlled or catalyzed reaction rates are much higher than the chemical catalytic reactions. Milder reaction conditions. If we take a chemically catalyzed reactions or a physically catalyzed reaction, they may require much higher temperature or much adverse pH conditions. But if we consider enzyme for the same reactions, it may possible to do the reactions in a much milder conditions. Like enzymatically catalyzed reactions occur under relatively mild conditions. That is the temperature below 100 degrees centigrade, like atmospheric pressure and nearly neutral pH. Because mainly whatever the enzymatic reactions occur, that basically occurs within our body. And within our body, our blood pH level is basically slightly alkaline, or our body pH level is nearly neutral. So that is why if a oxidation of glucose have to take place within our body, that oxidation must take place at a very milder conditions which suitable for our body. That is why whatever the reactions takes place or whatever the reactions catalyzed by the enzyme, that react each and every reaction must have a milder reaction conditions than the chemical catalysis. Greater reaction specificity. Enzymes have a vastly greater degree of specificity with respect to the identities of both their substrate, like reactants and the products, than do chemical catalysis. They have a specificity that which products they need to convert. Enzymes can differentiate between the glucose and fructose and galactose and each and every individual carbohydrates or the other biomolecules. So that is why they are very specific in their action. Enzymes are fastest catalytic molecule. They are very fast. Their activity is very fast. They are able to accelerate reaction by factor of as much as millions or million or more. Indeed, most reactions in biological system do not take place at perceptible rate in the absence of enzyme. A particular reaction in presence of enzyme and in absence of enzyme, their main difference is basically their rate of the reactions. A particular reactions may occur in absence of an enzyme, but that reactions may not have may not be that fast. So that is the main basic point of being a successful utilization of enzymes, or the main functionality of the enzymes. That is their acceleration rate. Even a reaction as simple as the hydration of carbon dioxide. The hydration of carbon dioxide, that means the association of water with carbon dioxide, is catalyzed by the enzyme. We all know the name of that enzyme, carbonic anhydrase. The transfer of carbon dioxide from tissue to blood and then to the air in the alveoli of the lungs would be less complete in the absence of this enzyme. That is why this enzyme is very important. This uh, as this Hydration of carbon dioxide is actually produce the bicarbonate molecules, and bicarbonate molecules is a basic thing which is actually used as a carrier of carbon dioxide. That means the carbon dioxide which is produced in our tissue, then carbon dioxide comes into the blood, and then through blood it comes into our alveoli, and then it removes into the air. So that thing is actually possible because of this enzyme named carbonic anhydrase. In fact, carbonic anhydrase is one of the fastest enzyme known. Each molecule can hydrate into the power 6 molecule of carbon dioxide per second. This catalyzed reaction is 10 to the power second 7 times as fast as the uncatalyzed one. So that is why enzymes are very much fast. They are able to accelerate the reaction rate. For enzymes, substrates are very important because enzymes are highly specific, both in the reactions as they catalyze and in their choice of reactants. And the choice of reactants, the reactant on which enzyme actually exerts its catalytic power, we call it a substrate. And enzyme usually catalyze a single chemical reaction or a set of closely related reactions. Like a particular enzyme named glucose, like a particular enzyme named hexokinase. Hexokinase enzyme is able to convert several kind of hexoses into its monophosphate form. Another enzyme named glucokinase or fructokinase, as its name suggests, they are able to convert only glucose and fructose into its monophosphate form. That means the enzyme, depending on the structure, depending on the location where they are actually working, their choice of reaction and their catalytic ability changes based on the reactants. That is why reactants are very important in case of an enzymatically catalyzed reaction. Let us consider a proteolytic enzyme, such as protease kind of enzyme. The enzyme is actually working on the proteins. In vivo, this enzyme catalyzes proteolysis, the hydrolysis of a peptide bond. Like this. This is a peptide bond, that pink and blue color bond between the pink carbon dioxide CO and a blue NH. CONH is a peptide bond. So the proteolytic enzymes are actually break this peptide bond by utilizing one water molecule. So these peptide bonds are actually basic building blocks of protein, but these proteolytic enzymes differ in their degree of substrate specificity. Like several kinds of proteolytic enzymes are present, like papain, which is found in papaya plants, is quite undiscriminating. It will cleave any peptide bond with little regard to the identity of the adjacent side chain. So they are not that much specific. If they find some peptide bond CONH group, CONH bond, they are actually able to cleave that. They don't bother what type of side chains are present or what type of adjacent side chains are actually present in the protein. The lack of specificity accounts for its use in meat tendering sauces. Whatever the meat tendering processes, they use papain. The digestive enzyme trypsin, on the other hand, is quite specific. Trypsin is quite specific. They need to have a specific carboxyl site of lysine and arginine residue. Otherwise, they do not exert their activity. Quite specific and catalyze the splitting of peptide bond only on the carboxyl side of lysine and arginine residue. They are only act on the carboxylic side of the protein, and only act in presence of lysine or arginine residues. So that, please subscribe the channel and hit the bell icon.